Okay, so hi everyone. First of all, I would like to thank you for your availability and your time today, for joining us in our webinar, our latest webinar at Fluigent and Secoya Technologies, entitled Controlled UV Crosslinked Microcapsule Production Using Microfluidic Technology. I'm pleased today to be joined by Marie Metler, our R&D engineer at Secoya Technologies, and Adrien Diond. It's the technologies lead at Secoya Technologies, and myself, Joseph Farah, microfluidic application engineer at Fluigent. So a quick for today. Our outline and our agenda for the webinar. We'll start by a quick introduction about microcapsules and general applications, followed by the presentation by the new UV-crosslinked microcapsule production platform with Adrien. And after, we will discuss with Marie the case study about the single crystals in microcapsule and how to generate them and do advanced study in this. And we'll close the session with a Q&A interactive session. And I encourage you, and please don't hesitate if you have any questions during the webinar to chat or to send us the questions on the Q&A panel that you can see on the bottom of the Zoom session. So with this webinar, we hope by the end of it that you will learn and discover how microcapsules are used nowadays as a versatile tool, either in drug delivery, encapsulation, crystallization, and so on. Explore the challenges of traditional microencapsulations methods that we are facing in research or even in some industry, and the potential of microfluidics that can help revolutionize this field. And after we'll discuss the advantages, and we'll focus on the advantages of especially UV-like cross-linking for rapid and solvent-free capsules production, with an insight about the new UV-crosslinked microcapsule platform with Secoya Technologies and the RayDrop and its advantages compared to classical microfluidic chips on the market. And finally, with the case study, we hope that you will learn how to use the microcapsules for the screening of crystallizations conditions. So let's dive in first about the microcapsules, quick introduction. So the microcapsules, they are the micrometric scale parts that can be particle, bubble, or liquid drops that is surrounded by the shell. The shell, which is generally, it can be organic or inorganic material, can protect what's encapsulated in the core material from the temperature, from the oxidation, the pressure, and overall environment of the capsule, which can afford some control release and design or engineer some targeted releases. This is why in the recent years, it's emerged as an attractive candidate for encapsulating, transporting, or contributively releasing a wide variety of technology of important active material. And this is why, and especially because of the advantages of the material that can be coated of the shell, either it is, which is generally a polymeric material. It has the advantages to be natural, available, renewable, and especially biocompatible and biodegradable, which opens several applications from especially the drug delivery, the food preservation, the wound dressing, or for example, the infection disease prevention. So we will go now with a couple of applications which can illustrate the advantages of the microcapsules in several fields. So first of all, one of the most or the major applications are microcapsules for drug delivery, and especially the microcapsules as drug carriers. So in this approach, the drugs can be encapsulated inside the microcapsules, and thanks to the properties or the characteristics of the shell, of the polymeric shell, it can either be dissolved like we can dissolve the shell, or thanks to some characteristics, it can release the drugs inside the environment that we are targeting in order to reach the target safely without being affected by the environment. And in this case, it can protect the drugs, control the release over a long time, which cannot be the case, for example, if we release directly the drug. The release of the drug can be not only based on the membranes chemistry, but also from an external stimulus. For example, from the temperature, magnetic fields, light, mechanical, electric, ultrasound, or enzymatic, or through the variation of the pH. And in this case, we can release what's encapsulated inside. And in this case, we open a wide variety of application, either by just applying these mechanical or external factors in order to control with high precisions the drugs or the encapsulated material. Another application or example using the microcapsules is the encapsulation for food preservation and nutrition. For example, in this application, we can encapsulate some minerals, vitamins, or probiotics inside the microcapsule, which can be added when wheat products, dairy products, or processed fruit products. And in this case, it can give the advantage of preventing unpleasant taste from fortified food, or prevent nutrition degradation or loss of due to the external factors. Another type of application is in the field of cosmetics. As you know, we can also encapsulate some core materials such as the skin moisturizing agents or skin cooling agents, vitamins, essential oil inside the wall shell, which can be, as we discussed, some natural polymers, semi-synthetic or synthetic polymers, and with doing some external factors such as pressure, friction, temperature, diffusion, it can release what is encapsulated inside. A quick simple example, it can be how to use, for example, for the skin. If we have the microcapsule, we can just pat it or rub it on the skin in order to release the core materials and eliminate the shell. And in this case, for example, it will be a great idea if we are encapsulating some volatile material or some precious materials inside the microcapsules. The microcapsules are not usually just can be used as drug carriers or just for encapsulations of materials that can be released after. It can be used also as an independent analysis vessels, as we can decrease from milliliter scale to picoliter scales. For example, we can encapsulate what we are needing to study, either some proteins or, for example, in drug discovery. And in this case, we can decrease hugely the amount of the samples that we want to analyze, and do the research within really reduced cost compared to traditional methods. One of the examples that we can explore is the drug delivery or the drug discovery. As you know, in order to determine or to discover a new drug, we usually target the protein structure. And as you know, the protein structure can be really complex, and we usually pass by X-ray analysis. And in this case, an application of the microcapsules can be to encapsulate the protein inside, to crystallize them, and do the X-ray analysis directly within just a few picoliters of the materials that we know. To illustrate more concretely, for example, as you can see, the protein structure that we won't target, or we will base in order to design the drug that we want. And in this case, we really need to discover its structure. And for this, for example, we can encapsulate it inside microcapsules and do the crystallization afterwards. And in this case, we can pass it to to accelerate the process by analyzing directly the X-rays through the X-ray analysis in order to reach the drug delivery or the drug discovery methods. However, in order to unlock all these applications, one of the key parameters is how to produce these microcapsules. And traditionally, what we want in this application, the microcapsules should be monodisperse with really controlled size, and of course, easy methods or efficient method in order to do these productions of these macrocapsules. Traditionally, the classics methods can be based on self-assembly, coaxial electrospray, gas shearing strategies, and recently it emerged as a microfluidics, the technology for these applications. Here I summarize a little bit the advantages and the inconvenience of each method. For the classical methods, such as the emulsifications or the drying-based methods, even though it is easy to put or to set up the experiments to do the production, but it's usually we don't have any control or really decreased control over the particle size, and usually it varies from batch to batch with high dispersity between the produced microcapsules. This is why the microfluidics emerged as a promising method, especially because we can control the size and the dispersity of each microcapsule that we want, and it's easy to create the double emulsion with it. However, it's still the instrument. It's really depending on the instruments, and we need some like the accessories or especially the microfluidic chip in order to reach these results of producing the microcapsules. As I told you, like even though we have the high monodispersity or high encapsulation efficiency, which can reach sometimes more than 95%, it's still difficult to handle, require in-house expertise and equipment in the microfluidic chip, and require specific surface treatment. As you know, the classical methods to use the microfluidics is to combine like two channels or sometimes like two microfluidic chips with the in order to create a first emulsion and after to create the second emulsion. And in this case, and lead afterwards to the microcapsules. Traditionally, with the classical materials, we need also some functionalization of the surface and some surface treatment in order to have this hydrophobic and hydrophilic. So all this inconvenience or this process can require a long time in order to reach them and decrease the opportunity of and the advantages of microfluidics. That's why usually when we want to adapt or to move into microfluidics chips or microfluidic techniques, we usually need to evaluate the performance of it. For example, the frequency, the size, the monodispersity that can lead the microfluidic chips, the phase configurations, the microstructures that we would like to create, and of course how to design the core and shell curing strategies afterwards. This is why in partnership with Secoya Technologies at Lyon, we'll present you now our offers in terms to solve these problems and to help you create directly and easily the microcapsules in microfluidics. Adrien?
Thank you, Joseph, for this comprehensive introduction to microcapsules and microfluidics. I will now introduce you to the Secoya Technologies for the formation of core shell capsules. So at Secoya, we were soon confronted to the limitation of a microfluidic device commercially available, rating issues, poor chemical resistance of the coatings, lack of robustness of the fittings. And that's why we finally developed our own devices called the RayDrop. And this device is based on the alignment in a metallic chamber of a 3D printed nozzle in glass or in resin in front of a collection capillary. Using this device, you can produce single emulsion droplets or double emulsion core shell type with a highly reproducible way. So with a high monodispersity, a high control on the size and on the size dispersion. I will now show you with a movie how it works inside the RayDrop. So the RayDrop is a device to produce single and double emulsion. It is based on the alignment in a metallic chamber of 2 inserts supporting 2 capillaries. One is equipped with an injection nozzle, and the other one is equipped with a glass capillary to collect the droplets. The injection nozzle is in glass or in resin. We first fill the main chamber with the continuous phase, then we connect the droplet phase to the nozzle, and finally the tubing to collect the emulsion. We'll now go inside the chamber to see how it works. At the junction of the nozzle tip and the collection capillary, the jet flowing out the nozzle is squeezed by the continuous phase, leading to the formation of very monodispersed droplets transported by the continuous phase at the outlet of the system. If we want now produce a double emulsion, we just have to change one insert, an insert able to carry 2 phases, the core and the shell phases, with the core being totally surrounded by the shell phase at the tip of this specific nozzle for double emulsion. And at the end, we collect double emulsion in the collection tubing out of the RayDrop. With this system, we don't face to limitation of the classical microfluidic chips, because there is no wetting issues, there is no contact between the droplets and the wall of the output capillary because of the cylindrical geometry of the system. So no coating is needed to obtain simple and double emulsion. Regarding the size that we can achieve with this system, we can go from 25 microns to 400 microns, depending on the geometry used. Actually, the size will depend on the geometry. By geometry, I mean the couple of injection nozzle and collection capillary. With one geometry, with one couple, you have access to one range of sizes. And to fine-tune the size in this range, you just have to vary the flow rates of the different fluids in your system. And if you want to change the range, you just have to change the 2 inserts on the main box to change the nozzle and the collection capillary. Keep in mind that the sizes here are the size of the droplets, but with some process, if there is shrinking of the polymer, you can obtain at the end of your process very small particles, let's say 10 microns, starting from 100 microns PLGA beads, by example, because there will be a shrinking of the droplets in the process, in the post-process at the output of the RayDrop. Now, as you probably know, a lab on a chip is often a chip on a lab, and it often requires expertise in microfluidics to operate the device. And finally, you will lost a lot of time focusing on this setup instead of focusing on your final application. That's why at Secoya, we develop a system fully integrated that integrates actually all is needed, all the equipment needed to operate the RayDrop. This system is called the RayDrop platform. On the left of this platform, you have all the controller, the pump from Fluigent, pressure controller based, the valve, the reservoir, and the filters. And on the right of the platform, between a camera and a light system, you have the RayDrop. So using this system, you have a fully integrated equipment to operate the RayDrop, and in a few days of training, you will be able to produce your own formulation in single and double emulsion, even if you're not used with microfluidics. To demonstrate the capabilities of this system, we produce droplets and double emulsion with the platform to produce beads and microcapsules. So by example, we produce beads of alginate, hydragid, but also PLGA, and also microcapsule with a shell of PLGA, chitosan, and the topic of today, microcapsule with a shell UV curable. So now I will focus on this application, how to produce a microcapsule with a shell made of a UV curable resin. To do that, we use a process called in situ process, meaning that the polymerization will occur in the tubing connected to the output of the RayDrop. This way, we avoid aggregation issue or coalescence issue that can arise if you collect first the droplet in reservoir and then apply the UV lamp on the system. I'm frozen. Sorry, I will restart my camera. Okay, that's better. So using this in situ process, we can go from liquid double emulsion fast to solid shell with a liquid core inside the particle. Then we develop a system to adapt this process to the platform. This is composed of a box in which the capillary connected to the output of the RayDrop passes through. On the cover of this box is the head of the UV lamp, and on the lateral sides of the box there are two windows with a UV filters to avoid that the user will be exposed to the UV radiation. So this system is totally compatible with the RayDrop platform, and using this system you can now produce microcapsules with UV curable resin, the most common being PEG microcapsules, PEG diacrylates, or gelatin metacrylate resins for cell encapsulation, drug encapsulation of whatsoever any application that you want. And another application is the production of polymetacrylate microcapsule for the study of crystallization. And I will now let the floor to Mary to explain to you how it works to study crystallization in microcapsules.
Thank you, Adrien. So for the case study, let's focus on the single crystal production. So the crystallization of molecules remain an important challenge, in particular for the X-ray diffraction analysis. For 20 years, microfluidics has been used to produce single crystals in highly controlled nanoliter volumes. First, we observed the crystallization in small chambers, and after that, with the improvement of the microfluidic, we developed droplets to make crystals inside, thanks to the diffusion phenomenon. Today, I will present you a new type of crystallization, which is based on the production of solid microcapsules. That will have the advantages to give a higher stability to the double emulsion and also thanks to the solid microcapsules. And it also enables a large choice of solvents. So the global representation of the crystallization process, as Adrien explained you just before, we use a RayDrop device, which is an emulsification device. And after that, the double emulsion produced is irradiated by the UV lamp in the UV module. Now I will give more example about the collect of the solid microcapsules and about the crystallization of the molecules that are contained inside the microcapsules. So we can adjust a lot of parameters as temperature, solvents, and time to have various crystals. And after that, we will present the X-ray analysis of the crystals contained in the microcapsules. First of all, the presentation of the system. So solid microcapsules are produced with the RayDrop, and the microcapsules are collected in a collection bath. The composition of the microcapsules is a solid polymetacrylate resin shell and a liquid core with a solution that will contain the molecule that you want to crystallize. We observed that the polymetacrylate resin shell is porous to solvent but impermeable to the solute. That's why we have a transfer through this membrane, and we observed that eventually we can have a nucleation of the molecules contained in the core, and in the best case, nucleation and crystallization will occur, leading to the observation of one single crystal or in many crystals in the solid microcapsules. You can observe that on the picture on the right. So one case study was the crystallization of copper sulfate in water as a core phase. The method that we developed is very useful for the screening of the parameters. There you can see that we collect capsules in various collection baths, for instance, containing acetone, ethanol, or also ethyl acetate. And we can also make a screening of the storage condition in temperature. So we collected various samples, and there you have 4 pictures of 4 different samples in different collection baths. You see that depending on the conditions, you will have different types of crystals. And there, the best conditions that were found were in the condition 3, so when we collect the microcapsules in ethyl acetate. So to make to put in a nutshell, there we produce solid microcapsules in polymetacrylate resin, and the core phase was composed of a copper sulfate solution. We collected the capsules in a collection bath of ethyl acetate and let this sample for 2 days at room temperature to let the diffusion occur through the solid shell and to create the nucleation. After these 2 days, we observed copper sulfate encapsulated crystals in the 6% of all the capsules of the sample. So there we have a crystallization which is caused by the antisolvent diffusion. Another example is the crystallization of the glycine. There we encapsulated a glycine solution in the solid microcapsules. The capsules were collected in the air, and we let the sample dry for 1 day at 4.5 degrees. After this day, we observed that 2.5% of the capsules were containing single crystals. There you can see a picture of the sample, and you can also see that we observed 2 polymorphs of the same molecule, which is the glycine, in the same sample. So this method is very useful because you can produce a lot of capsules in very few seconds. And in the sample, you can also make a screening to observe and after that analyze various polymorphisms of our molecule. Another example is the crystallization of protein. So there we focused on the lysozyme crystals, and we encapsulated a solution composed of sodium chloride solution with lysozyme. We encapsulated this solution and collected it in a sodium chloride solution to have some exchanges through the porous membrane. After 3 days at 4.5 degrees Celsius, we observed encapsulated crystals of lysozyme, as you can see on this picture. There, the crystallization is caused by the water diffusion through the shell. And by producing various samples and collecting in different collection baths, we observed that depending on the concentration of sodium chloride in the collection bath, we can have an improvement of the production of single lysozyme crystals in 1 microcapsules. That will be useful for X-ray diffraction analysis. Second protein that was studied was the proteinase K. So there, we encapsulated the proteinase K solution and collected it in a collection bath composed of an ammonium sulfate solution. After 2 days at 4.5 degrees Celsius, we also observed monocrystals of the proteinase K in capsules, as you can see on this picture. There, we also have production of crystals thanks to the water diffusion. So what can we do when we have our protein or molecule to crystallize that is crystallized in the capsules? Then we can store the sample and we can ship the sample, because thanks to the solid microcapsule structure, we can ship the sample easily, and we can make an X-ray analysis on the capsules. So we have two methods. You can either make the analysis in situ, so you let the structure of the microcapsule and you put the microcapsule containing the crystal to analyze in a mounting loop. After that, the analysis can be performed because the microcapsules is totally transparent to the X-ray irradiation. Either, if you want to collect the crystal, you can break the shell with a tweezer, for instance, and you can collect the crystal to make a direct X-ray analysis on the crystal. As a conclusion, at Secoya, we developed a method to produce and to make crystallized molecules inside solid microcapsules. This method allows us to produce 2,500 independent capsules per minute. So this is a huge production, and it is not resource consuming, as you only have to dissolve nanograms of solutes for one capsule. So you only need a very small amount of solute for an experiment. This is an easy screening method, as I underlined before, thanks to the fast parameter tuning and to the collection bath use. And the crystals are encapsulated in a solid shell that allows the protection of the crystal, but it is also compatible with the X-ray analysis and also with low temperature freezing. Then the shell is transparent, that will help for the direct X-ray analysis too. So all these results are now published in the chemical communication. Thanks. I would like to thank also the University of Namur that did the analysis with us and helped us for the interpretation of the results, and the Secoya team which worked on this application. And the perspective of this method for the production of solid microcapsules with crystals could be to make some guest infusion for the co-crystallization of some API. We would like to work on the sample preparation for serial X-ray diffraction, and huge shell exploration could also be done by studying the chemistry, the porosity, and also the thickness of the shell. We can also study the use of organic solvent as a core phase to make crystallize some molecules that are only dissolved in some organic solvent. So thank you, and now I will let Joseph give you a small conclusion.
Thank you, Maria and Adrien, for these explanations. Well, just for the conclusion. So as we discovered in this webinar, using the RayDrop based microfluidic chip and all the systems in terms of flow controllers and valves can make easier the production for microcapsules, more practical. And in this case, in our case, it can open the way for several applications, from the production of beads such as alginate, PLGA, which we previously discussed through previous webinars or on our sites. And we complete our packages or offers from the PLGA, chitosan, PMMA, or PEGDA microcapsules, or through also the FACS platforms that we have in terms of encapsulating biological materials. There are just a few examples that can be done with the RayDrop and its platform, but of course all microfluidic systems can be replicated into double emulsions processes in order to create your own application. So thank you for your attention and for your presence in this webinar. And now we'll be happy to have some questions if you are from you. So I encourage you to ask us through the Q&A panel that we have. Maybe I will start with the first one. We have the range. For example, the range of the droplets or the microcapsules that we can create with the RayDrop.
So that will depend on the geometry of the nozzle, but typically we can produce droplets from 25 micrometers to 400 of micrometers. And that will also depend on the formulation that you use. If, for instance, you use a PLGA, you will have a huge solvent extraction, so you will have a shrinking of your particle after the production.
Okay. Another questions we have from Ludovic. Do you have recipes to tune finely and predictively the shell thickness depending on the flow rate, interfacial tension, and viscosity, or it is quite empirical, the user having to test a bit randomly before finding the good parameters?
I would say both. There exists a lot of models available on publication. And we have a model for the size of the droplets in single emulsion published about the RayDrop. But now, to my knowledge, it's quite many empirical to find the right parameters for your application and your formulation, that the best way to achieve your final goal. But yeah, I think that it definitely exists some models, but I'm not sure that it will be easy to be applied directly to this system. And generally, in this kind of models, you need to have a lot of information about your formulation regarding the interfacial tension, regarding yes, and that can vary due to the addition of surfactants. That's not easy stuff actually. So mainly we do that empirically. Yes.
Another questions: can the capsules afterwards be processed in microfluidics, for example, for sorting purposes?
Yeah, actually, we have an application and an application note about the use of the platform RayDrop to encapsulate cells in an oil shell, a fluorinated oil, to be used then in post-process in a FACS system, so in a sorting system. So in this case, the encapsulation is a preparation for this post-process in FACS sorting.
We have another question which is also related to FACS. Is it possible to sort it? We encourage you to check on our websites, and even we can discuss more the FACS applications that we have. We already developed the parameters that we need to use in order to have how to do the sorting with the FACS machines.
I can just give a fast answer to this question.
You know?
The main criterion is the size, actually, because the nozzle of the FACS can accommodate some sizes, small sizes. That's why we developed this contour nozzle to be able to produce very small double emulsion droplets down to 20-25 microns.
Yeah.
And a question from Jeremy: what is the wavelength of the UV light that was used during the applications?
There it was 285 nanometer. Yes, nanometer. And the wavelength has to be compatible with the photo initiator. So there, I didn't spoke about that, but when I use a polymetallic resin, we also mix this resin with 0.1% of photo initiator that will react to the UV light to make the double emulsion solidify and to create the solid shell. So there, the wavelength has to be compatible with the photo initiator that you will use.
Mm-hmm. And we can adapt the system to different UV head to be compatible with your photo initiator. So there exist the different heads for different wavelengths, actually.
Another question, which is which size variation have you observed during the application of the microcapsules?
So that will depend on the formulation. There, for the polymetallic resin, you don't have size variation during the process. So between the double emulsion and the solid microcapsules, we have a low CV, so the polydispersity of the sample is very low. It's usually under 2%.
Which is the best compound to work with cells for about 20 cells in a drop? That it relates also some into some fax applications.
Yeah.
Yes. Yeah. There is. Yeah. I don't know exactly about the formulation for this application. For what?
I think we use HFE oil for the shell, and the water it was used as a core and as a continuous phase. Of course, you have to work with phases that are biocompatible. So usually it's the oil that is used for the cell encapsulation.
200 cells in per drop is a very high amount, I think, and it's difficult to achieve. You will definitely need to use what we call the Baker geometry, so the nozzle to produce droplets, double emulsion with a core or about 200 microns. And be very careful for the homogeneity of your cell solutions to avoid clogging if some cells aggregate at the nozzle tip. Yeah, usually we encapsulate, let's say, about 50, maximum 100 cells per droplets. But yes, it will definitely depend on your cell solutions.
And also on the fact that if you want to sort the cells after in a fax or not, because if you just want to encapsulate the cells without doing sorting after, you can make a bigger double emulsion.
Again, don't hesitate to contact us if you are really interested in fax applications in order to discuss more with it. Back to the microcapsules, which chemistries create the most homogeneous cells during the experiments. It appears in our images that some shells differ in thicknesses.
The shell is normally quite homogeneous between the capsules. Again, the homogeneity of the shell will depend on the formulation. So which chemistry is the best is, I would say, maybe the chemistry that don't have a solvent diffusion. For instance, if you make some PLGA, maybe you can have some difference in the shell homogeneity. But normally with the other formulation, you don't have a lot of not homogeneous shell.
What you can observe in the images we showed is a difference in the shell thickness from batch to batch, from image to image. But in one batch, in one production, the shell are very similar. Because we use the same parameters, but from batch to batch, we can change some fluidic parameters, some flow rate, and then we have some variation. But in one production batch, the size of the shell is very similar. Yeah.
Is it possible to use lipids to obtain solid lipid microcapsules?
I don't know. Yes, we can use lipids, but in this case, more to we have an application note about the formulation of lipid nanoparticles using lipid. In this case, we use the RayDrop to form a co-flow, so to mix both fluids, the aqueous phase and the lipid phase, in a diffusion only way. And in this case, we observe the formation of lipid nanoparticles, but to obtain lipid microcapsules, we have no experience about this. But yes, actually, all what is described in literature using microfluidics device for single and double emulsion can be done in the RayDrop. Maybe there is some exception, but usually that is the rule. What is described using a flow focusing chip, let's say in glass or PDMS, can be done with the RayDrop.
Another question about the compatibility with organic solvents of the RayDrop.
Yes, we have, as I said, we have both nozzle available, one in resin and one in glass. The nozzle in resin is not compatible with dichloromethane and other solvent in the same family, like chloroform. But in this case, if you want to use this kind of solvent, we have the option with the glass nozzle. The other materials are stainless steels and glass, so there is no issue with classical solvent, I would say.
Another question from Tao is about the UV exposure time in order to do the curing of the microcapsules.
Yeah, that's a very, very short time. The capsules flow inside the collection capillary, and it's just like a flash. So just the time that the double emulsion go under the light, that will be enough to have a solid microcapsules after. So it's less than 1 second per droplet.
Question from Virgil: is it what specific meta-acrylate-based resin did you use?
In fact, we don't have the exact formulation of the resin because it was a company, it's a commercial resin. But yeah, we know that it's a polymetacrylate based resin, and we also did some tests with another polymer, so with PEGDA. So it's also working with some PEG based polymers. So we only made a few samples with the PEG formulation, but it can also work with other formulation.
Another question from Ulysse: what is the volume range of the two solutions injected for the double emulsion?
That will depend on the flow rate that you will use. For my part, I usually work with a flow rate of 10 microliters per minute for the core phase, and around 20 microliters per minute for the shell phase. And we didn't speak about that, but we also have the possibility to add an injection loop on the platform. And for this case study, it's very useful so that you have your injection loop on the core phase and you just have to inject 20 microliter of the solution with the molecule that you want to crystallize, and you can collect the capsules containing this solution. So when you use the injection loop, the volume will be very, very low. You can go up to 20 microliter, and you have also, of course, some bigger volumes.
What is the smallest particle size that can be achieved?
Yeah, we already discussed about this topic. You have to make a difference between the particle and the droplets. Actually, the droplets, I would say 20-25 microns, but particles will depend on the post-process, solvent extraction or UV curing. And so we can go down 10 microns in the PLGA case, or be limited to 25 if there is no shrinking. It depends.
A question from Jeremy: after the shell polymerization by UV, is it possible to open the microcapsules?
Yeah, of course, you can open the microcapsules when you tweeze it. So you use a tweezer and you take the capsule and you just splash it. So you can break it in small parts. And after, if you want to collect the crystals, it's totally doable.
And
there was a question about the maximum number of droplets that can be generated, but it is feasible to produce approximately 100 million droplets. So yes, if you have the time, there is no problem because it's a continuous system.
Yeah.
So let's say it will depend on the flow rate, on the frequency of generation, and this frequency of generation of droplets will depend on the geometry of the system and the flow rates. But typically, it's between let's say 10 and 1,000 of hertz, so kilohertz. So yes, if you let the system operate, you can produce 100 million droplets. Yes, no problem.
A question from Barbara. We discussed a little bit. Maybe we can discuss more about it. How reproducible is the size under the same conditions? The size of the droplets or the microcapsules?
Yeah, it's very reproducible because you can fix the flow rate, and when you fix the flow rate, you will have the same size at the output of the redrop. So you can produce the same experiment more times. When you use the same flow rate, you will have the same size at the output.
Typically, we measure a coefficient of variation less than 2%, and in most cases, less than 1%. That is the typical value of microfluidics, actually, because we are working on a dripping regime. So this regime of droplet formation in simple and double emulsion gives the best result in terms of size dispersion of the droplets than the particles.
Another, there are a lot of questions about if we can share the paper. Of course, after the webinar, we will have the replay of it, of the presentations that we did, and also we'll share the references in terms of the papers and all the material and informations needed. And don't hesitate to contact us again in order to have more information if you have any inquiries or interest in the or any additional questions. Yeah, I think we can we'll end up the webinar with this. For any additional questions, don't hesitate to contact us. Thank you, Marie and Adrien.
And thank you, Joseph.
And thank you all for your participation in this webinar. And see you soon in new webinars. Thank you.
Thank you.
Bye.
Have a good day. Bye.